Good morning. Good morning to all of you. Good morning, children. Today our topic meristematic tissue. You know about the types of tissues in plant body. The type of tissues in plant body are permanent tissues and meristematic tissues. The cell which retains the capacity to divide are called meristematic tissue, and the cell which loses the capacity to divide are called permanent tissues. Meristematic tissues are developed from embryonal tissue. A group of meristems is called meristematic tissue. That means the cells of meristematic tissue are called meristems. A group of meristems is called meristematic tissue. Every meristem is a immature cell, evergreen, living cells with the power of division. Meristems are immature cells. That means never undergo maturation. They are evergreen young cells. Evergreen young cell means they never undergo maturation. So simply they are called evergreen young cells. Evergreen young cells and living cells with the power of cell division. A meristem never contains a suberized wall. Suberized walls are completely absent in meristematic cells. Means the cell wall of meristem has only two layers in their structure. The two layers are, one is the middle lamella and second one is the primary cell wall. Secondary cell walls are completely absent. So only the secondary cell wall made up with the suberin or cutin type of materials. So the secondary cell wall is absent and suberized walls are absent. Only the cell wall of meristems are having two layers in their structure. The two are, one is the middle lamella and the second one is the primary cell wall. Primary meristematic tissues are developed from embryonic tissues. Embryonic tissue means you know about embryo. The embryo has two types of parts. One is the embryonal axis and second one are the cotyledons. This embryonal axis has two tips. One is the radicle and second one is the plumule. Plumule develops into a shoot system. Radicle develops into a root system. But both of these two, plumule, radicle, are divides by mitosis and form meristematic tissues at their tips, form root apical meristem and shoot apical meristem. So all primary tissues, almost all the primary tissues are developed from embryo. So embryonic meristems. They developed from embryonic meristems. Meristematic activity are shown by only apical regions, leaf primordia and intercalary meristem and birds. Apical region means you know about shoot apex and root apex. So both shoot apex and root apex are two important apical regions of plant axis. Leaf primordia means the leaf which are developed from a group of meristematic cells. Simply it is called leaf primordia. So apical regions, leaf primordia divides by mitosis. So they are example of meristematic cells. And intercalary meristem also present at the place of node. Even birds like axillary bird and terminal bird. Axillary bird and terminal bird are two important kinds of birds that are also having some amount of meristematic tissue. Birds are develops into inflorescence or develops into a lateral branch. So we see the meristematic activity in apical regions, leaf primordia, intercalary meristem and in birds. Based on position, meristems are divided into three types. The three types are: one is a apical meristem, second one intercalary meristem, and the third one is lateral meristem. Apical meristems are present at apices. They are present at shoot tip and root tip, apex of root and apex of shoot. The meristematic tissue which is present at shoot tip is called shoot apical meristem. Root tip is called root apical meristem. The meristematic tissue which is present at shoot apical meristem, root apical meristem. Simply they are called SAM and RAM. Shoot apical meristem and root apical meristem. Intercalary meristem is present at the place of node. Node means one swollen structure. For suppose if we see any one plant axis, this is an internode and it is a node. Below node also one internode is present. This is an internode. Here in this internode, we see this swollen structure node. Here inside the node, we see a meristematic tissue. Here this meristematic tissue is called intercalary meristem. So intercalary meristem present at the place of a node between permanent tissues. And lateral side of axis, we see another type of meristematic tissue. The type of meristematic tissue is called lateral meristems. All lateral meristems are cylindrical meristems. All lateral meristems are cylindrical meristems. The next one. The position of RAM is terminal and the position of SAM is subterminal. RAM and SAM, root apical meristem and shoot apical meristem. Primary growth of plant body occurs by apical meristems and intercalary meristem only, not by lateral meristems. Intercalary meristem and apical meristems are involved in primary growth of plant body. And lateral meristems are involved in secondary growth. So all cylindrical meristems are responsible for secondary growth of plant body. Apical meristems are absent at the tips of leaves. That means for suppose if we see any one leaf like this, here this is a tip of leaf. Here there is no meristematic tissue. So meristematic tissues are absent at the tip of leaves. Lateral meristems are active. The lateral meristem means cylindrical meristems. Lateral meristems are active only in mature stems and roots of dicots and gymnosperms. Why? Because in monocot plant, we can't see the lateral meristem. Any part means that is a stem or a root or leaf or etc. So in monocots, the lateral meristems are completely absent. Even in underground parts also, meristems are absent. Not in underground parts, sorry. Even in leaves, even in the leaves of all. In the leaves of all, after a certain growth, in leaves of all plant like monocots, dicots and gymnosperms, meristematic tissue is completely absent. But a part of meristematic tissue present at its axil in the form of axillary bud. Lateral meristems are completely absent in parts of monocots. You remember, all the secondary meristems are lateral in position. All the secondary meristems are lateral in position, but not secondary in origin. Because you know about intrafascicular cambium. Intrafascicular cambium also called fascicular vesicular cambium. Intrafascicular cambium also called fascicular vesicular cambium. This is a lateral meristem present at lateral position, but it is a primary meristem. So all lateral meristems are not secondary meristems. All secondary meristems are not lateral meristems. Why? Because intrafascicular cambium is lateral in position, but it is a primary meristem. Remaining two lateral meristems are secondary meristems like cork cambium and interfascicular cambium. So you see secondary meristems like interfascicular cambium and cork cambium are only two secondary meristems and lateral in position. All the secondary meristems are formed from permanent tissues by a process called de-differentiation process at the time of secondary growth. At the time of secondary growth only, the permanent tissues regains the capacity to divide and differentiates into secondary meristems. So secondary meristems are formed from permanent tissues by de-differentiation process. Primary meristem increases the length of plant axis. So the plant axis increases in its length by secondary meristem or primary meristem. Only primary meristem. But increases in girth of plant or width of plant by secondary meristems. By secondary meristems. Mainly by two different type of secondary meristems. The two are, one is the vesicular cambium and second one is the cork cambium. So both vesicular cambium and cork cambium are responsible for increased girth or increased width of root and stem in dicots and gymnosperms. Intercalary meristem present in majority of monocots, but not in all. But not in all. Again, I repeat the statement. Intercalary meristem present in majority of monocots. 99% of monocots, the stem axis may be divisible into nodes and internodes. At the place of node, we see the presence of intercalary meristem. So intercalary meristem is present in almost all monocots, but not all. Axillary buds are developed from shoot-apical meristem, not from intercalary meristem. Why? Because for suppose if we see this is a one vertical axis. Here in this vertical axis, this is a leaf. You know about that, the axillary buds which are present at the axil of leaf at the place of node. Because it is a node place now. So axillary bud presented near to the node, near to the intercalary meristem, but it is developed from shoot-apical meristem, not from intercalary meristem. Very, very, very important in NEET point of view. Axillary buds are developed from shoot-apical meristem, not from intercalary meristem. But axillary buds are present at the place of node. They are present at the place of node in the axil of leaf. For suppose if they want to ask a question as, how many shoot-apical meristems are present in a twig of plant possessing 2 branches and 18 leaves? For suppose if they want to ask a question like this, simply the answer is 3. Why? Because how many are the branches? 2 na? For suppose this is a one axis. It has one branch in right side, another branch in left side. It also terminates with one shoot apex, and here also one, and here also one. So how many are the total shoot apical meristems are present in this axis? 1, 2, 3. So 2, 3. So 3 are the shoot apical meristems which are present in a plant with 2 branches, because 2 SAMs are at tips of branches, one at main axis, one at the tip of main axis. So how many are the total shoot apical meristems present in a twig? 3. A primary meristem that produces secondary tissues is fascicular cambium. You know about the other name of fascicular cambium as intrafascicular cambium. So that intrafascicular cambium, also called a fascicular cambium, it is a primary meristem according to its origin. But it involves in the development of secondary tissues at the time of secondary growth, like secondary xylem, secondary phloem. All of that are formed from fascicular cambium. So a primary meristem that produces secondary tissues is fascicular cambium or intrafascicular cambium. Intercalary meristem present in between permanent tissues. Already we know about that. So at the place of node, that means for suppose if we see this is a node. At the place of node, intercalary meristem located here. But around the intercalary meristem, who are present? Permanent tissues. Permanent tissues. So intercalary meristem present in between permanent tissues. In grass plants, if any part injured, not only in grass plant, in majority of all other grass plants near to the node, all that parts are developed from, or injured parts are developed from, or repaired by lateral meristems. Repaired with the help of lateral meristems in plants. Intercalary meristem involves in the regeneration of parts of grasses removed by grazing animals. You know about that grazing animals are herbivores. When herbivores are eat grass from a grassland, then again and again, grass plants are produces from their nodes. Branches are produced from their nodes. If you see the characteristic features of meristems, the meristems are thin-walled cells. The walls are very thin. And they are having prominent nucleus and dense cytoplasm with more mitochondria. Why more mitochondria are needed in meristems? Because to divide a cell into daughter cells or to maintain the dividing capacity, there is a need of a huge amount of energy. That huge amount of energy produces by the presence of more mitochondria. Intercellular spaces are absent in between cells of meristematic cells. These are always immature cells. Based on function, the meristems are divided into three types. That are, one is a protoderm, ground meristem, and procambia. Protoderma involves in the formation of dermal tissues. Dermal tissues are protective tissues that are included under a tissue system. The name of tissue system is called epidermal tissue system. And the second type of second region in shoot apex and root apex is the ground meristem. It involves in the formation of ground tissue system. Procambium involves in the formation of vesicular tissue system. So vesicular tissues are developed from procambium, ground tissues are developed from ground meristem, and dermal tissues are developed from protoderm. These three are the regions in shoot apex and root apex. So if you see any one shoot apex and root apex, in both shoot apex and root apex, we see these three regions. The three regions may appears like this, one inside the other. That means it is a protoderm, and this one is a ground meristem, and the central one is a procambium. So the three regions are present inside the shoot apex and root apex. In shoot apex and root apex, we see these three regions that are protoderm, ground meristem, and procambia that are involved in the formation of three different tissue systems of plant body. All the permanent tissues are developed from meristems by two different type of processes. That are, one is a differentiation process, and the second one, redifferentiation process. You know about the differentiation process means primary meristematic tissue involves in mitotic division, form two cells. One cell loses its capacity to divide and develops into a primary permanent cell. A group of primary permanent cells is called primary permanent tissue that are present in primary plant body. All these primary permanent tissues are formed by differentiation process, not dedifferentiation, differentiation process. Again, the secondary meristematic tissues are loses their capacity to divide and differentiates into secondary permanent tissues by a process called redifferentiation. So all permanent tissues are developed from meristems by differentiation and de-differentiation process. The type of meristem present in a vesicular bundle of dicot stem is primary meristem. The name of that primary meristem is intrafascicular cambium. So intrafascicular cambium is a primary meristematic tissue which is present in between xylem and phloem of vesicular bundle. A cambium is always lateral in position, but it is either primary or secondary in origin. Correct sequence of appearance of various meristems of a woody dicot stem during its lifespan are: one is the apical meristems, first formed meristems. After that, fascicular cambium and interfascicular cambium. Lastly, cork cambium. All secondary meristems are formed from simple permanent tissues like parenchyma and collenchyma. Mostly from parenchymatous tissue, also from collenchymatous tissue. Rarely, collenchyma also develops into meristem, but most of the secondary meristems are developed from parenchymatous tissue. You know about that cork cambium is one important secondary meristem. This secondary meristem developed from parenchyma of cortex. And one more important interfascicular cambium. Interfascicular cambium also another type of secondary meristem. This interfascicular cambium developed from parenchyma of medullary race. So mostly secondary meristems are developed from parenchyma, also from collenchyma, right? Thank you. Thanks to one and all. This is the information about parenchymatous tissue. Meristematic tissues. A complete information about the meristematic tissues. Right.